Hi, my name is Andrew Murray. I am a faculty member at the Department of Molecular and Cellular Biology and the Center for Systems Biology at Harvard University. And my plan today is to give you a talk about sex, not in its full gory X-rated version, but the sort of sex that a model organism, the budding yeast Saccharomyces cerevisiae, carries out. This talk is a general introduction. It's going to last about half an hour. There will be a longer talk which gives more detail and talks about how scientists run through and discriminate between different possible explanations of how something works by doing experiments. So to get started, there's a famous saying, a loaf of bread, a jug of wine, and thou. But in fact, everyone except thou is provided by our friend the budding yeast. So here is a pitcher of beer. Here are 2 bottles of wine. If you look carefully, this says Andrew Murray Vineyards. That's not me. That's just someone who happens to have the same name and work on the same organism, but in a slightly more productive way. And here is the loaf of bread. And here is the guy who produces it. So these are budding yeast cells. They're about 5,000ths of a millimeter. That's 5 microns in diameter. And as you can see from this guy, right down here, they divide by budding. So there is mom, and here, right there, is her little daughter. And so these are the guys we're going to talk about. And you might want to ask, why work on something crazy like this when we can study sex in humans in bedrooms and stuff like that? And the answer is very simple. People are complicated and yeast are simple. And I'm just going to go through Peeps versus Yeasty. So these are the Latin names. We're Homo sapiens. The actors of today's drama are called Saccharomyces cerevisiae. And here are some differences. Okay? It takes about 20 years to double a human being. It takes 90 minutes, way shorter, to double a yeast cell. We're made up of about 50 trillion cells. So you started out with the fusion of your mother's egg and your dad's sperm, and then there have been many, many, many billions and billions of cell divisions to make you. A yeast cell is a single organism. So each time the cell divides, that's 2 new organisms. So everything is simpler, faster. We have about 6 billion bases of DNA in each one of our cells. If you're a yeast cell, it's only 12, so it's about 500-fold less DNA. It's about 5 or 6-fold fewer genes. But here's the big deal. There's only so much experimentation you can do on humans. We're not allowed to do forced matings on anyone. We're not allowed to do genetic manipulation. In yeast, the bottom line is anything goes. That means genetic manipulation. It means forced mating. And it means a version of Survivor that you will never see on Fox, where almost everyone dies and only the fittest survive. So we can actually look at evolution in the laboratory in this organism. And last but not least, everyone has sex, including yeast. And so what we're interested in doing is understanding how cells find each other to mate in this much simpler system. So to talk you through that, I have to start out with the very basics of the cell division cycle, the process by which you turn one cell into 2. So here we go. This is the diagram of the simplest possible way of imagining the cell. So the circle here is the outline of the cell. This line is a chromosome, and that little black dot in the middle is a specialized part of the chromosome that's going to attach to the machinery that will ultimately separate 2 sister chromosomes from each other. So the first thing you have to do is you have to replicate your DNA. So now there are 2 chromosomes here where there used to be 1. Then you need some sort of machinery to separate the chromosomes from each other. So we're going to generate 2 genetically identical daughter cells. And last but not least, the cell's going to have to divide. So here we are back at the beginning of the cell cycle. But now instead of 1 cell, we have 1, 2. Okay? And one of the things that you'll notice is that during this whole process, and this is true of almost every cell division, but not quite all of them, these cells are growing and getting bigger so that the daughters that get produced are the same size that their moms were when they were born. All right? So now what we're going to do, that's the simplest possible cartoon. So now we're going to do 90 minutes in the life of our friend the budding yeast. So we're just going to walk our way around a single cell cycle. So here's a yeast cell down on the bottom. It's an ovoid cell. It has a nucleus, which is highlighted here. The cell's going to grow, get bigger, and it's going to ask a bunch of questions at this point. So those questions include, am I big enough to be allowed to keep going? Have I got food? And in particular, the thing that we'll talk a little bit about today, are there any partners? Can I smell anyone of the opposite sex around? And the reason that this happens only once in this cycle is when two cells fuse with each other, you want them both to have the same number of chromosomes. So if one of the two cells had replicated its chromosomes and the other hadn't, you'd have some weird mixed up cell which would be unable to decide which chromosome should get replicated and which not. So in budding yeast, mating occurs at one point in the cell cycle, this period, as the cells are born and growing, but before they get up here, where they switch from growing uniformly, just blowing up like a beach ball, to growing non-uniformly, because here's the little bud pooching out, okay? And it's before this happens, this part down here is the only time when you can mate, okay? We get bigger, we set up the machinery to segregate the chromosomes, but before we segregate them, we get to ask another question: are my chromosomes all lined up properly? Because if they're not, I'm not going to make genetically identical daughters. If they are, we can break one nucleus into two, the cell divides, and we're all the way around again, okay? So the next thing we want to talk about, so that was the cell division cycle of one cell. One yeast makes two yeasts. Now we want to go through the whole life cycle. If it was people, this would be like taking your family tree and starting with mom and pop, going to you, meeting someone cute, getting married, having children, and so on. So there are two mating types, the equivalent of our two sexes, male and female, but in yeast they're just called A and alpha. And this is a haploid cell, meaning it has 16 chromosomes, one of each sort, and it's going to bud, get bigger, and divide, just like I showed you on the last slide. And here is an alpha haploid. All it differs in is a single small piece of DNA on chromosome 3, so there are no X and Y chromosomes. And these cells, the alpha cells, make a perfume called alpha factor, and the A cells have a receptor that detects that pheromone. The A cells make their pheromone or perfume, which is called A factor, and the alpha cells here detect that A factor and respond to it. And when they respond, the cells don't bud, but they make these little protuberants. They grow in a non-uniform way. So this is a different sort of non-uniform growth from budding. And these little things are called shmoos. And I'll tell you where that name comes from in a second. Their tips touch, they fuse. This is the first diploid cell, so now we have 32 chromosomes, 16 from momish yeast and 16 from popish yeast. And these cells can once again divide. They can go around this cell division cycle like that, and they can keep doing that again and again and again, unless you starve them, in which case they go through the specialized cell divisions that are called meiosis. They make 4 spores, 2 A's and 2 alphas, and if you feed them again, these spores will germinate, and we get right back to the beginning again. So that's the whole life cycle, and it's this part here, this business of mating, that we're interested in. Okay? And so what I'm going to do now is to show you a night at the yeast single spar, and what we're going to see, this is a movie, and there are 2 cells. Here's an A cell, and here's an alpha cell. You're going to watch them grow towards each other, meet in the middle here. They're going to fuse, and they're going to produce their first daughter right over here. So here we go. You watch them grow and grow and grow, and they're going to touch, boom, and here comes daughter right there. Okay? And I'll play that for you one more time. Here we go. We want to watch this guy grow, and we're going to see fusion right there, and there comes the daughter. Okay? That whole process there, which you saw, takes about an hour and a half in real life, so it's been speeded up a lot. So what I want to do now for you is define different sorts of ways that cells move when they feel chemical gradients. So the first, which is you might be more familiar with, is something called chemotaxis. That means that the whole cell moves just the way I'm walking towards the screen, towards some gradient. And I can give you some examples of that. So one of them is a bacterium swimming up or down a gradient of attractants. If it smells sugar, for example, it can swim up a gradient of increasing sugar concentration. If they get inside your body, they can swim in various directions. We don't like bacteria inside our bodies, and so there are specialized cells called neutrophils. And what they do, they're some of the white blood cells in our body, is they literally hone in on the vapor trail of waste products that the bacteria leave, which are different from the sorts of waste products that our bodies make, and they literally zoom around and catch the bacteria. And last but not least, boy moths can smell from astonishingly long distances, on the order of a mile, the pheromones, the perfumes that are emitted by female moths, and they can fly up the gradients of those concentrations to mate with each other. Okay? So that's distinct from what scientists call chemotropism, and in chemotropism, it's cells that can't move. So a yeast cell doesn't have legs, it doesn't have a propeller, it can't crawl. The cells just sit there, but what they can do is they can do non-uniform growth. So they're growing predominantly in the direction of some chemical gradients, and some examples of that, as you were developing inside your mother and as you grew up even, neurons that were born in your body, they had to send out processes to find things like muscles to go from your spinal cord to your brain. And when they do that, they also follow gradients of chemicals, but they're growing, not moving. In plant sex, you have a plant flower and an insect lands and it deposits pollen on it. The pollen, which is like a sperm in some sense, but it doesn't move, it sends out a tube, and that tube has to grow and grow and grow and find the plant equivalent of the egg. It's technically called an ovule, and that's how you get fertilization in plants. And we're going to talk about, and I just showed you in the movie, yeast sex, where A and alpha cells grow towards each other, their tips touch, and they fuse. So that reminds me to say something important, which is this is an example of what biologists call symmetry breaking, a stage where cells are growing uniformly, like blowing up a beach ball. It's the same shape, it's just getting bigger and bigger. And that's what happens just after a baby yeast cell has been born, and it's growing bigger and bigger. Eventually, when it gets big enough, it's allowed to do non-uniform growth, which means that on this point, right on the tip of this cell here, a little part of the cell will pooch out because the wall that surrounds the cell has gotten softer, and this is how you make the bud, and the bud grows bigger like this. And during mating, what happens is you have a similar sort of non-uniform growth, but now, instead of a bud, the cell grows out this tip, and the tip gets bigger as it grows towards its prospective partner. And the reason they're called shmoos is there's a cartoon strip called Little Abner, and there were creatures in this strip which were called shmoos. They look a little bit like a mating yeast cell. They had certain interesting properties. According to the shmoo legend, the lovable creature laid eggs, gave milk, and died of sheer ecstasy when looked at with hunger. Okay? And so one of the things I want to talk about, and in the talk that goes into more details about how testing ideas works, I can come back to, is which direction cells pick to shmoo in. So this is a diagram, it's a cartoon. Here's a gradient of pheromone, low at the top, high at the bottom. Here are 5 cells. When they make shmoos, they will make them incredibly reliably in the direction of higher pheromone concentrations. So this is a classical example of chemotropism. You're growing down a concentration gradient of something you see as an attractant. But here are 5 cells, and instead of being put in a gradient of pheromone, the pheromone concentration is the same everywhere here. But even those cells will eventually break symmetry and they'll make shmoos. They'll make them in all sorts of random directions. And this is, like I said, an example of something called symmetry breaking, which happens very often in biology. It's not particularly well understood, and one of the things that we're interested in studying. Okay? And so to help put the framework for things, I want to point out the differences between chemotaxis and mating. So if you are a bacteria swimming up a concentration gradient, or you're one of your white blood cells chasing that bacteria and trying to move faster, things happen fast. Cells move several times the length of their own bodies within a second, whereas mating is slow. That movie I showed you took place over 2 hours. So it's sort of slow, stately courtship, a bit like courtly love in the 13th century. Chemotaxis is a long-range process. If you're a male moth and there's a female moth half a mile away, you have to find that concentration gradient and you have to keep flying up it for a long, long time. Whereas mating is a profoundly local process, a little bit like villages in the 15th and 16th and 17th, 18th centuries. Unless 2 yeast cells are less than 1 cell diameter apart, they don't even know each other exist. The pheromone concentration is too low. And the last thing that's different is when cells mate with each other, they have to pick a target. If you imagine yourself, or someone you know, in a bar, sitting on a bar stool, flanked by 2 very attractive people of whatever sex you prefer, and you're drinking, at the end of the evening, the goal is to turn towards one of them or the other and not to just topple drunken off the bar stool exactly halfway in between them. In the end, you have to pick a target. And there are lots and lots of examples in biology, especially in what's called developmental biology, the question of how you build organisms that involve this sort of picking a target. And we see this as a simple example of a general class of problems. Okay? So now I have to tell you how we study how cells polarize, and so we're going to start right over here. This is a yeast cell that hasn't yet budded. And we've colored some things. This red thing is actually telling the cell where it should bud. It hasn't budded yet, but we can tell from this red color that it is going to bud there, and that's because we've marked something called the polarisome with a fluorescent protein. So we can see that fluorescent protein underneath the microscope. Behind it, in the green color here, there's a ring of stuff laid down which helps to strengthen the cell wall except where the bud is going to pooch out. The proteins that make that up are called septins, so we can label them in a different color. And last but not least, these straight lines that you see going back here are actin cables. So they're made up, they're polymers of a small protein called actin, and they are the cell's railroad tracks. So stuff gets transported by motors, which are seriously analogous to locomotives, and it will come down this railroad track here, be deposited here, and this is the new material that's used to grow cell membrane and cell walls. And last but not least, and not labeled here, the blue thing is the nucleus of the cell where the DNA is. And so we can watch how these markers behave, in this case as we go around the circle this way, as the cell buds. So as I told you, that accumulation of red, the polarisome, tells you where the cell's going to bud. And later on when the cell's going to divide, right down here, those same polarity proteins, and especially the septin ring, is required in order for the cell to divide successfully. Those same proteins get recycled, if you like, when cells are treated with pheromones. So this is an example of a cell that's been treated with alpha factor, so you're fooling an A cell into believing that an alpha cell is somewhere nearby. And here's the tip of the shmoo, which is going to grow up like this. The polarisome is right there. There's a distribution of septins behind it, which is required to keep that cell wall strong behind the growing tip so that the cell just doesn't blow up like a beach ball. And the actin cables stream back from the tip of the cell, because this is all the stuff that's going to make new cell. It's going to go up these filaments and get deposited here so that the cell can grow. So now what I'm going to do, and this is probably going to be the hardest part to follow, and I will try and go slowly, is give you a molecular view of how all this signaling machinery works. You can still get a lot out of this talk if you don't follow all these little details. So here we go. The most important thing to say is this is the outside of the cell, this is the inside of the cell here, and right here, this railroad track, this gray line with the thicker gray lines on top, is the plasma membrane. This is what separates the inside from the outside. Here comes alpha factor, this little black horseshoe. It's going to bind to this protein that snakes back and forth through the membrane, the alpha factor receptor. And that protein in turn is going to talk to this protein here. This is what's called a G protein. And it's called a G because it stands for guanine nucleotide binding. And what you can see where it says GDP is this molecule has a molecule of guanine nucleotide diphosphate bound to it. So what's going to happen? The pheromone binds to the receptor. The receptor changes its shape. Its change in shape causes it to interact physically with this G protein, and it makes the G protein let go of its GDP. So the GDP comes off. That's diphosphate. GTP, guanine nucleotide triphosphate, comes on. And that causes a change in the structure of one of the three subunits, the one called alpha subunit. You can think of it as its surface getting all bumpy. The beta and gamma subunits float off, and they're going to be the business end of how the cell communicates from the outside to the inside that it's seen the mating factor. And the way they do that is they recruit scaffold proteins. So this light blue color is a scaffold protein. It's called FAR1. FAR stands for pheromone arrest. So this was found by geneticists. CDC42 stands for cell division cycle. So this was found by people working on how the cell cycle works. This protein stimulates indirectly the formation of those actin filaments that are going to drag everything the cell needs to the surface. Okay? It does something else. It activates a class of molecules called protein kinases This one is called sterile 20. Again, geneticists call these sterile because mutations in these genes keep cells from mating. This is a class of protein called a protein kinase. It chemically modifies and affects the activity of other proteins. So sterile 20 modifies sterile 11, which in turn modifies sterile 7, which in turn modifies this guy called Fuz3, and this all ultimately leads to gene transcription, and it's responsible for arresting the cell cycle so that the two cells that mate with each other are in the same part of the cell cycle. Okay? So there's still some more stuff to go. So one of the things that's important is for the cell to polarize in one direction, to break symmetry, something has to differentiate some parts of the cell surface from other parts of the cell surface. And to do that successfully, the rich have to get richer and the poor have to get poorer. This is a little bit like certain governmental philosophies in our country. And these green arrows indicate that what a biologist would call a positive feedback loop. So if we follow the arrows around, CDC42 stimulates actin polymerization. Everything that's associated with the membrane, for example, the pheromone receptor itself, has to be delivered to the surface of the cell along these filaments. So the more filaments you have, the more of this molecule you deliver. The more of this molecule you have, the more G protein you activate, the more CDC42 gets turned on, the more actin you make, the more pheromone receptor you get, and you keep going around this circle as things rev up, basically. And so it can cause little random fluctuations to get amplified into big differences between different parts of the cell. So that's how you turn things on. You also need, at the end of the day, to turn things off. So to give you an analogy, your visual system adapts to the ambient light intensity. So if you walk into a dark room, you get more sensitive to dim light. If you walk out of a movie theater in the middle of the afternoon into the blinding California sunlight, your visual system, everything temporarily looks blindingly bright, and you adapt. If you didn't adapt and recover, you would still be looking at the blinding white light of the delivery room that a doctor put you in. Okay? So you need ways to turn things off. So one way to turn things off is this protein, which is called SST2. So that's the SST stands for super sensitive to pheromone, because these mutations don't recover well from seeing pheromone. And what this does is it hydrolyzes, or helps to hydrolyze this GTP back to GDP. The bumps go away on this alpha subunit. It binds back to the beta and gamma subunits. The scaffolds fall off and signaling stops. You also need a way, if your courtship partner has disappeared or gets too far away, to turn things off. And in particular, you need to deal with the fact there's still this alpha factor on the outside of the cell. So if you're an A cell, you make this molecule here called bar1, and bar1 is drawn as a little Pac-Man here. And what this is what's called a protease, a protein that cuts other proteins, and it literally cuts the alpha factor in half so that A cells can recover from being exposed to alpha factor if they don't mate successfully. Okay? So what we want to do is ask in detail how cells respond to pheromone gradients. And to do that, we have to make gradients that are reproducible, whose structure we know about. And we do that using something called microfluidics. And so I'm going to show you the top view and a cross-section of a microfluidic chamber. So this is looking from the top. The bottom, behind the screen, would be a microscope slide. The top, closer to you, would be a cover slip. Cells would be glued on top. And what you can see is when you look down, fluid comes in two streams. This one has alpha factor. And this one here, which goes up in this way, doesn't have alpha factor. Because of the dimensions of the device, this is only about 1 millimeter across, and the speed of the flow, there's no turbulent mixing. Anything that's going to move from one side of the channel to the other has to do it by diffusion. What that means is this blue wash, which is the pheromone, gradually gets more and more spread out across the gradient as you go down, and at each point in the gradient we can draw a cross-section, and we can plot the concentration of the pheromone as you go across that cross-section. And you can see as you go from the top of the device and you flow down, the gradient just gets shallower and shallower. So that's looking from the top. If we cut it like this and look at a cross-section, here's our microscope looking down at the cells inside. Here are the cells that are glued to the cover slip. And this is the liquid that's flowing. It would be flowing right out of the screen towards you in this image. Okay? So what I'm going to show you is what happens to cells in such a chamber. So in exactly the same way as it was on the left side, the flow is going to be in this direction here. This is the high concentration of pheromone on this side of the channel. This is the low concentration. When I run the movie, what you're going to see is each time the movie loops round, these white circles will draw your attention to a different bunch of cells. First, you'll see cells that don't shmoo. They make buds, but they make the buds towards higher pheromone concentrations. Then you'll see a group of cells over here that just get bigger and bigger like basketballs, so they're still growing symmetrically. The cells around here are going to make shmoos. They're going to head straight up the gradient. Then there are going to be some cells here which start in the wrong direction and then turn. And the guys at the top at the highest pheromone concentration are going to be hopelessly confused. Okay? They're not going to know which direction to go in. So here we go. So here you see the guys bud straight up the gradient. Here are the beach balls. Here's a cell that's going to shmoo straight up the gradient. Here's a cell that's going to turn up the gradient. And these are the hopelessly confused. Okay? So I'll let it loop through one more time. And what you're going to see is these guys are going to bud straight up the gradient. These are the beach balls right here. Cells shmooing straight up the gradient towards an imaginary partner. And here are cells turning up the gradient. And these guys are the hopelessly confused. So I'm going to leave it there. There's another slightly longer talk that goes into much more detail. It goes into trying to understand how it is that cells break symmetry and how it is that yeast cells can mate successfully that vary under conditions which in a human analogy would be a man and a woman on a desert island, but no one else, and the crowdiest, sweatiest college mixer you've ever been in with thousands of people of two opposite sexes. And at the end of that, yeast cells are so good at finding partners that almost no one goes home at the end of the night brokenhearted and crying. And if you want to see that, keep watching.